my name is Joseph Milken, and today we're going to be talking about a specific macromolecule, proteins. In the last video, we talked about the 4 different types of proteins, macromolecules, and we talked about carbohydrates and lipids. And we're specifically going to talk about proteins. Why are proteins important? Well, according to Dr. Dini at the Texas Tech University, proteins, actually, it's not even just him. If I use a good pen, proteins. The root, proteo, means first. Why does it mean first? Well, it's not because it's the first in line or whatever. It's because proteo, for first, means most important. So I'm going to put importance. Why is it? Well, because think about it this way. So you might say, well, DNA is the most important part of cells. I mean, the most important macromolecule, I'm talking about nucleic acids. And you can make a claim for that, but the proteins are what moves it does all the functions for all the DNA. Whether that is from the metabolism with enzymes, or you're talking about transport proteins. Proteins do a lot of stuff. I mean, obviously, we just talked about carbohydrates and lipids, and the main functions of those are structure and energy. But how did the energy get there? How did the hormones get to move, outside the cell for, signaling and so forth? That's from proteins, and that's what they mean by proteo, first, okay? So like we said in the last video, the monomers of proteins are amino acids. Okay, I'm going to let you look at that real quick. Let you draw it if you want. Okay, we drew the lipids in the last episode, so we have amino acids. Okay, so at the very center of the amino acid, we have a carbon. Okay. And we have a carbon attached to that. We have a hydrogen attached to the other side. We even have a hydrogen attached to the top of it. And below this, we have R. What is R? Well, R is an R group, or a functional group. And if you're in organic chemistry, there's a whole list of R groups that you have to memorize. But for this class, it's not necessary. Okay? And I think I told you wrong. This right here is actually N for nitrogen. Okay? Right here we have another hydrogen, and then right here we have a double bond with oxygen to the carbon on the right side, and then we have an OH group. Now, these, we just talked about functional groups with R groups, okay? Well, these are R groups, okay? So we might as well learn the name of this, okay? This right here with the nitrogen is called an amine, okay? And this right here on the right side with a carbon with a double bonded oxygen and a hydroxide, this is called carbolics. I always screw up with it. Carboxylic acid, something like that. Okay? Basically, double bond oxygen with a hydroxide connected to a carbon. Okay? That's all you need to know. Okay? And I'm not going to talk, if you want to go and watch Dr. Dini's video, he talks about the exception being cysteine, but he didn't talk about it in lecture, so I'm not going to go over it. Why is this important? Well, let's talk about bonding, okay? Because obviously, if we're going to learn about the structures of carbohydrates, lipids, nucleic acids, and proteins, we might as well learn about how they bond together. I mean, because they're not all just one macromolecule. When we're talking about macromolecules, usually they're, bonded together in big groups. We talked about polysaccharides. So, just like we did in the last one, I'm going to erase this real quick, and I'm just going to draw this end right here. I have a hydrogen and a hydrogen connected to a nitrogen. We call this the amine group. Okay, and I'm going to bond the hydrogen, the hydroxide together, just like we did with the glucose, and that's going to create H2O. And obviously, just like in the last one, we're going to form a bond between the carbon and the nitrogen, and this right here is going to be a very strong covalent bond. Okay, obviously, because it's got to be able to stand being in an aqueous solution, and we called the bonds for not lipids, for amino acids to be phospho diester or ester bonds. Or I'm just going to put bond singular because there's only one bond. Okay, that's how they're going to bond. Okay. I'm sorry, I told you wrong. Not a phosphodiester, duh. Because we remember that the monomer for protein is a amino acid, but the polymer is a polypeptide. The bond right here is not a phosphodiester. That's for amino acid. This is actually a peptide bond. Sorry about that. Okay, but we live and learn, right? Now the thing about this is that these bonds right here are prone to turning. Okay, so like you see that I got these arrows right here. This means that the molecule, like if I'm looking, and it might be helpful to see like a 3D diagram of this. Well, let's say I got the amine group, okay? And this right here, the nitrogen can spin on its bond, okay? If it had a double bond, that would be a different story. Like this oxygen is not going to be able to turn. Like for example, let's say that I have these two pens right here, okay? I can be able to spin it, you know, like because it's just one bond. There, you know, it's able to spin like this, you know. Let me show you specifically. I'm able to turn it like that. But if I have two, I'm not going to be able to I'm not going to be able to spin like that. I'd have to be able to spin both. I can't turn it like that. That's not what I'm talking about. So, since they're single bonds, they're able to rotate. Okay. Why is that important? Well, if you learn in organic chemistry, that's going to create torsional strain, which will increase the energy needed to break that bond, making it even stronger. Okay. We talked about the water being taken away in order to polymerize these macromolecules, and again, this process is called dehydration synthesis. Okay, you're going to be hearing that a lot. We've already said it 2 times, both for lipids and for carbohydrates, but we're talking about proteins here. Okay. Most proteins that perform functions are called polypeptides. Poly meaning many. So obviously more than one. Okay. Right here, but the same rules apply that we had for carbohydrates. So if you want to go back to the video and look at that, I'm just going to talk about it. An oligo protein being one with around 10 or more proteins. A polypeptide, I said it wrong, didn't I? An oligopeptide being 10 or more, around that proteins, with 50 or more proteins / peptides being a polypeptide.
It would rotate with the carbon.
What I should have done is, like, if you look at your fingers, you can twist your fingers like that. But if I have 2 fingers together, I can't twist it like that. You know what I'm saying? Like, the bond is stronger, but there's something about being able to twist molecules on there that it's called torsional strain. And that increases the electricity, the energy needed to break that bond. Okay. Obviously, I mean, since it's a covalent bond, they're still very strong bonds. Let's talk about the functions of polypeptides / proteins. Okay. Actually, first, let's talk about there's 2 different types of polypeptides. So we're going to go polypeptide. And there's two different structural forms. You can have a linear, and you can have a globular. What do I mean by that? Well, remember, if you remember from talking about the extracellular matrix, when we talked about intermediate filaments, we talked about microtubules, and we talked about microfilaments. Remember that we talked about with actin, which is the structural protein of microfilaments. So remember that there's like F actin and G actin. Okay, so I got F actin, F being filament, and this is, and just to clarify, this is the protein, the structural protein of microfilaments. Okay, to G actin, G being globular. Okay. Let's say that I'm going from F, which are the filaments, which are the tubes, to globular, which is, like globs, obviously broken up filaments. This is called, you guessed it, depolymerization. I'm going to do depolymer. What if I want to turn globular into filament? So I'm taking a bunch of different globs and making it into a filament, so where you can actually transport different macromolecules or micromolecules across the cell. Like, for instance, when we're doing the secretory pathway and we're wanting to transport different hormones or messengers like mRNA to different parts of the cell. Well, in order to do that, in order to create that microfilament as it goes on, transport it using the myosin, we polymerize, or polymer, I'm just going to put polymer, okay? And that's what we're talking about right here, those two different types, the microfilament, or the F-actin, being the linear, and then the globular being the G-actin, or the globular protein of microfilaments. So hopefully you're able to make that connection to what we've already learned about in the class. Hopefully you learned it the first time. Let's talk about an example of a linear protein. Okay, you might have heard of this. Collagen. Okay, collagen is a linear polypeptide. Okay, now what are collagens used for? Well, they're in tendons. And tendons are used for strengthening. It's a strengthening protein, and they actually have 3 chains of polypeptide in collagen. Let me clarify something real quick. Let me going back to the structure of proteins. If you go back and look at it, hopefully you've drawn it on your notes and you can look at it. Remember that we had that carbon connected to the amine group and the carboxylic acid. Again, I can't pronounce it very well. That R group right there is not included in what we call the backbone. Anything that is not connected to the R group / the R group is considered the backbone of that polypeptide or peptide. And the reason why is because the R group is where you're going to do the polymerization reaction, what we call the dehydration reaction. Okay? Don't quote me on that. But I do know for a fact the R group and anything connected to the R group is not considered part of the backbone of the protein. Okay? Let's talk about proteins and the 4 levels of protein. Okay? Proteins have 4 different levels of structure. Okay? And we're gonna go from smallest to largest. Okay? Which is kind of backwards to me, but it's just how it is. Okay? The smallest structure we call primary. Okay, I'm actually gonna draw it as 1 degree, and I'm gonna write primary. Okay, well, what does primary make up? Well, this is the structure of amino acids. Remember that the monomers for proteins are amino acids. So primary would be the structure, the multiple amino acids in order to create this protein. What about the second level? Well, by the way, just to make sure you know, this second level is optional. Okay, so it may or may not be in the protein structure, but it's important to know. The secondary level, okay, what does that make up? Well, this is regions where hydrophobic meaning they don't like water. Okay, remember that in a phospholipid bilayer, the inside of the bilayer is where the the hydrophobic tails are located. Remember, so hydrophilic heads are touching the water, and the hydrophobic are not touching the aqueous solution on the outside. Okay, regions where hydrophobic amino acids predominate. What do I mean by that? Well, let's say that I have this structure called an alpha helix. Okay, this is a type of protein, or a structure of protein. Okay, and let's say that I got like these two different protein backbones. Actually, these are not protein backbones. Sorry. I have this like spring-like structure, this being the protein, and then these are held together by hydrogen bonds. Now, you might be saying, whoa, wait a minute. We just talked about covalent bonds. Well, hydrogen bonds are not as strong as covalent bonds, okay? But when there's thousands, hundreds, thousands, even millions of hydrogen bonds, this structure is indeed a very strong structure. However, one thing to note is that while these hydrogen bonds are indeed strong when they're together, these bonds are prone to break, okay, which will lead to a flex in that spring, okay, what we call the alpha helix. What about well, if there's an alpha helix, well, there's got to be a beta something, right? Well, yeah. We call that, I'm going to draw it right here. This is called a beta pleated, like your pants, sheath. Is it sheets or sheets? I have sheets, okay? I'm going to draw it like this. So I got like this kind of long deal held together by hydrogen bonds. Okay. What is important to know about secondary? Well, in an aqueous solution, so I'm going to write here, aqueous, AQ. I know that doesn't look great, but secondary, or I'm going to write second degree, is generally found in the interior of protein. Okay, so we're still on the inside of the macromolecule, and that makes sense. Well, the inside of the protein is not touching the aqueous solution, so that's a hydrophobic area. So that's where the secondary structure is going to be. Okay. Remember that in this structure, those weak bonds the hydrogen bonds are weak and unstable, and while together they're strong, they're still individually weak and unstable. So you're gonna see that, like, for example, let's draw out the alpha helix structure, molecular-wise. Okay, so I got a nitrogen, I have a carbon, and then I have another carbon with another nitrogen here, and I'm kind of drawing the roundabout shape right now. Okay, I got another carbon. I got like about 2 nitrogens for every carbon. Okay, now this is linked up to a hydrogen. Okay, and there was a double bonded oxygen connected to this carbon up here. Okay, how are how are these going to bond together? You obviously see that these are held together. The sides from just splaying out. They got to be held in that helix shape. Which is like right here. So how are these going to hold together? Well, they're going to be bonded. Okay, but I'm going to draw a dashed line because these bonds can break and form again. Okay, and if that oxygen is going to steal the hydrogen from the nitrogen in order to keep the shape. But as you're constantly breaking and forming that bond again, you're changing the shape a little bit. So there is going to be a little bit of flexibility. However, in terms of the whole helix shape, since there's so many hydrogen bonds, you're going to maintain that shape. It's never all at once going to break all the hydrogen bonds. But it's important to note that especially since oxygen, and I know I'm trying that horrible, are both very electronegative. Okay, so obviously they're going to be pulling at this hydrogen. That's why that bond's going to be breaking. Okay, they're constantly pulling at each other. Okay, draw that if you want. So if you follow the pattern, the third level is going to be tertiary. And this is not an optional level. This is a legit level. And again, I'm going to draw it degree, third degree. Okay, what is that? Well, this is the 3D structure of a single polypeptide. Okay. So primary and secondary have led to the formation of a peptide. Well, what about, and so now we've formed up one peptide with another peptide, or if we're talking about polypeptides, we're actually going to be talking about 50 or more peptides. So I have like 50 peptides come together, and that 3D structure is the tertiary structure. So secondary being the linkage between these individual peptides, and then the primary being the formation of one peptide. Okay, maybe I should draw that out to help better illustrate that. Okay, so first one is peptide, one peptide, and it's just and I'm going to write in parentheses individual atoms. Okay. The second level, and is an optional level, is peptides bonded together. I take it back because these peptides, it's still technically, I screwed up. So let me explain. Remember that we said that secondary level of polypeptides are still in the hydrophobic area. So technically, it's just inside of the hydrophobic, or inside of protein, which is in turn a hydrophobic area. Okay. that tertiary level is a bunch of polypeptides, or I'm sorry, a bunch of peptides bonded together, okay? So peptide to peptide. And by the way, I'm going to write times 50, just to remember that it's a polypeptide, and polypeptides have 50 or more proteins. So now we've talked about tertiary. Well, you're like, well, one step up. That's right. Quaternary. I'm going to write that right here, or fourth degree. I know I didn't spell that right, but please forgive me. This is a hard word, okay? Quaternary is the 3D structure when more than one polypeptide associate. Okay, so now that we've formed one polypeptide with like 50 different peptides, or amino acids specifically, because that's what the monomer is. So 50 amino acids. Let's make sure we're using the right phrase. 50 amino acids to make a polypeptide. And now we have 50 and 50. I mean, those are arbitrary numbers, but 50 or more amino acids in a polypeptide, more than one polypeptide associating with each other. So now we're 4. Can you see that? Yes. It's poly. I'm going to just write poly to poly. Okay. Okay. So in case any of that was really confusing, primary and secondary are the formation of an amino acid. Okay? The individual atoms and the hydrophobic areas of the polypeptide, of the amino acid. The tertiary level, now that we've formed a amino acid, is the amino acid to amino acid in order to form one polypeptide. And then finally, quaternary is polypeptide associating with polypeptide. Okay? Let's draw out a quaternary level of protein. Okay, so I have this whole jumble. It's a whole chain. Okay, and then I got on another end. This is gonna link up somewhere. I'm just gonna say right here. Okay, another polypeptide. Okay, and by the way, the polypeptides being one single strand of protein, okay? Not globular, it's a strand, it's linear, okay? Well, how are these quaternary level structures being held together? This is why this is not part of the backbone. I think I was right about it earlier, okay? So let me clarify. Remember that we showed that these polypeptides, the linear versions of the polypeptides, when they, or the amino acids, when they bond together, they're using they're forming covalent bonds or peptide bonds between each other. But what are these amino acid chains, the polypeptides, using to hold each other together? They're being held together by R group interactions. Remember I said that R group is not a part of the backbone of amino acids. That's because these are what are holding together these polypeptides. And guess what? That is it. One thing to know. Let's say that I have I have this little beginning. I know that might be hard. You're going to have an N terminus And let's say that I got the start, the finale of this right here. Okay, this is going to be the C terminus. Okay, and being the actually C being the beginning, N being the end terminus, if I remember correctly. Of course, I don't remember it. I think I'm fine on that. Another thing to keep in mind when we're talking about that quaternary level, how do these form up and how do these break? Again, through R group interactions. Okay, so we have this. I know it looks like a blob, but really it's just one big, large chain of amino acid or polypeptides. We can denature it to where we only have, we can break down those polypeptide bonds to where it's just one, like one chain, okay? And then technically we have two chains. I know, haha, that's funny. I know you're laughing about two chains. We have a black, we have a blue. I get it, I have two blue here, but just know that we're breaking down that the polypeptide, or the R group interaction / polypeptide bond in between that. And same thing, we can also renature it to form, okay? Okay. I think that is it for proteins. So I hope this video helped. If you have any questions, just leave a comment or message me, and I'd be more than happy to either make a correction or to answer your question. One thing that's important to note about tertiary structure is that when those bonds form between the amino acid and the amino acid, these bonds are formed at random. Now, why is that important? Well, the structure of the protein is what dictates its function. So when I break the weak hydrogen bonds and they form at a different point on the structure, this changes the function of the protein because every single structure has a different function.